Hi, and welcome to Foundation Frontiers. In this educational series, we'll be discussing the application of comprehensive genomic profiling for detection of oncogenic fusions using two complementary approaches, DNA and RNA sequencing with Foundation 1 CDX and Foundation 1 RNA. To review, there are four types of DNA alterations detected by comprehensive genomic profiling. These include substitutions, insertions and deletions, copy number alterations, and the focus of our discussion today, rearrangements. Rearrangements include large chromosomal events, including deletions, duplications, inversions, and translocations. Here we're looking at an example of an inversion on chromosome 2, which leads to the fusion of EML4 and ALK. This produces an oncogenic protein. Fusions and rearrangements have been targeted in multiple types of solid tumors. Some of these include non-small cell lung cancer, which is known to be enriched in oncogenic fusions. Here we think about targeting RET, ALK, and ROS1. Importantly, there are also pan-tumor fusions. Those include NTRK 1, 2, and 3, as well as RET fusions, which are present at low levels across various tumor types. When we're thinking about gene fusions, it's important to consider where these occur and how they can be detected. So many gene fusions occur in the intronic regions of DNA. So these are in the non-coding regions of DNA. And when this happens in a common chromosomal breakpoint, they can be readily detected by DNA sequencing. However, when these occur either in an uncommon region of the DNA or when they occur in a large repetitive intron, they can be more difficult to detect with DNA-based NGS. A famous example of that is NRG1, which is a gene where fusions do occur. However, it has large repetitive introns, making this very difficult to detect in DNA-based NGS testing. Because the introns have been spliced out of RNA sequencing can help overcome this limitation of DNA sequencing by only sequencing the spliced exonic regions. Here we're looking at some of the pros and cons of DNA and RNA-based fusion detection. DNA-based NGS, we know, can be used to detect common fusions such as those in EML4 ALK or FGFR2 fusions. We have high quality DNA material in most cases, and this allows us to sequence and have accurate fusion detection. However, it is less sensitive for some of these oncogenic fusions, such as those involving genes with large repetitive introns, such as NRG1, which we discussed earlier. RNA-based NGS does overcome this limitation of DNA-based NGS by sequencing across spliced exons. It can help discriminate splicing isoforms, and it can also clarify the functional significance of fusions by only identifying those that are expressed. However, we do know that RNA-based NGS has a high failure rate, with anywhere between 9 and 50% failure rate being reported in the literature. The Foundation Medicine Foundation 1 CDX bait set has been designed to enable DNA-based detection of rearrangements and fusions. 13% of the sequencing region has been dedicated to intronic coverage to enable detection of these events. We also utilize a hybrid capture-based approach, which allows for rare and novel fusion partners to be detected, as only one gene in a rearrangement must be baited for detection. And the last thing is that we do use a de novo assembly approach, which allows for precise breakpoint detection. Here, we're not mapping the reads to the human genome, which means that those reads that are not easily mapped to the human genome representing fusions are not discarded and can be detected. Even with this well-designed DNA-based baiting approach, RNA fusion detection can still add supplemental benefit. So here we're looking at the results of a modeling study where we saw an estimated 1.3% overall RNA-only fusion detection rate in non-small cell lung cancer patients. This went up to 4.4% when we looked at samples that were otherwise driver negative. So adding RNA to an already well-designed DNA CGP can have this additional benefit. In order to detect those fusions that are not easily baited by a DNA NGS assay, we've added Foundation 1 RNA. Foundation 1 RNA is a laboratory-developed test that's been available since May of 2024. This is a supplemental offering to FoundationOne CDX for detection of gene fusions and rearrangements from RNA in solid tumors. There are 318 genes analyzed in this test. And importantly, there's no additional specimen required in addition to what's required for FoundationOne CDX testing because of a co-extraction process. And for reporting, you'll get one integrated test report. This provides clinicians with clinically actionable information for treatment selection. Next, we'll take a look at a hypothetical patient case where we can demonstrate the utility of RNA sequencing. So here we have our patient who is a 58-year-old male non-smoker and presents with a persistent cough that does not improve with treatment. He has an X-ray and subsequent imaging reveal a lung mass indicative of non-small cell lung cancer. A high-quality tissue sample is collected. So would this patient be a candidate for DNA and RNA-based NGS testing? So knowing that this patient has non-small cell lung cancer, we do know that this is a tumor type that's enriched in potentially actionable fusions. We see a low prevalence of NTRK fusions, RET fusions, ROS1 fusions, as well as ALK fusions in this tumor type. We also know that our patient is relatively young and a non-smoker, so this is a phenotype that might be enriched for these actionable fusions. And here we're looking at the result of this patient's NGS testing. FoundationOne CDX sequencing was completed. FoundationOne RNA sequencing was also completed. And you can see here that an EML4 ALK fusion was detected by both DNA and RNA sequencing. This allows the patient to become a candidate for ALK-targeted therapies. Some key takeaways of our discussion today. FoundationOne CDX is able to detect actionable fusions through selecting intronic coverage of DNA breakpoints and sequencing common fusion partners. RNA sequencing does add a supplemental benefit to this well-designed CGP assay, and FoundationOne is a supplemental offering to FoundationOne CDX, which can be used for detection of cancer-related fusions and rearrangements. Thank you for joining us today, and we're looking forward to seeing you on future Foundation Frontiers.